So this next talk is by Pascale Machaut. She's a senior researcher and team leader at the Centre National de la Recherche Scientifique in Marseille. And she had a connection to Palmer. She was here for a sabbatical some time back, I think she's going to mention. But it was very influential, and she's a common guest back to the campus as a result. She's going to be speaking on common determinants in natural toxins and non-toxin molecules to recognize acetylcholine binding macromolecules.
Thank you very much, Mr. Chairman. I'm very pleased and honored to be here again, I would say, because I was here 20 years ago for 2 years, as mentioned just now, as a sabbatical visitor. And since then, I came back several occasions on the campus to work with Palmer and several of his people. So, hopefully I will try to review some of the works we made together during these years. And I just want to stress out something which, during the preparation of my talk, I decided to change a little bit. So, the new title is a little bit different, and here you can read it. But, main part of the talk is going to be the same, the same ideas. And the time when I was in, how does it work? It's frozen. Okay.
Try it again. There we go.
Thank you. Okay, so this is a very old slide. It is 20 years ago, 20 years old. At that time, I think we had very good apparatus, cameras, but that slide was very overlight. And I'm very happy to see that in that slide, we were not so many people at that time in the lab, but at least 5 of these people are here in the room today. And I think this is very nice for us to remember all these times. And when I came to the Taylor lab in 1994, I brought several things to the lab, I think, with me in my luggage. And the first thing I brought about this was my salary, which was very comfortable for Palmer because he didn't have to pay me. And the second thing I brought about was my husband. And my husband is a crystallographer. And I think that was a very nice opportunity for us because we set up crystallography and crystallogenesis in the Taylor lab at that time. And I'm very happy to see how many other people afterwards used crystallography to solve very nice structures of cholinergic proteins in the Taylor lab and other proteins as well. And the other thing I brought about was fasciculin, because I was raised as a biochemist and a pharmacologist. And in my former life, during my PhD training, I used to purify and analyze pharmacologically snake toxins from snake venom. And I had the fasciculin in hand, and I came to this lab because I decided to come to the Taylor lab because I knew that in that lab I could find, nicotinic receptor to acetylcholine and acetylcholinesterase. And I was very interested into using acetylcholinesterase to know better how my fasciculin molecule would be able to inhibit the enzyme and to work afterward. So the first thing we were able to do at that time was to combine what we knew of the fasciculin molecule, the structure of which was solved just before by ourselves in collaboration with Juan Fontecilla, who is here. And we were able to define when solving this structure the differences between this molecule, which is an inhibitor of acetylcholinesterase, and very homologous other molecules with 3 fingers, which were also found in snake venoms, which were just alpha-neurotoxins, which were known at that time for inhibiting, as antagonists of the nicotinic receptor to acetylcholine. And we were interested in knowing, what were the determinants which was common to this kind of toxins, to make them very well-folded molecules with 3 fingers, and what were the determinants which were specific to these kinds of toxins to make them either antagonists of the nicotinic receptor or inhibitor of acetylcholinesterase. And at that time also, we knew that fasciculin was a very cationic molecule, which was full of positively charged side chain, and we already suspected that the mode of action of fasciculin was related to these cationic properties, because that was just after the solving of the structure of acetylcholinesterase was U.S. Sussman and his collaborators, as just mentioned previously, and the definition of the peripheral ionic site, where we knew that fasciculin would bind. So we were very happy with my fasciculin molecule purified from the venom by the milligram amount, and the acetylcholinesterase recombinant enzyme made in PalmerTaylor's lab at that time, to be able to crack the structure of this complex of acetylcholinesterase and fasciculin at the very same time as Yoel and his co-workers made with the torpedo enzyme, as Yoel mentioned earlier. And the only thing I want to stress out on this picture is that fasciculin binds at the entrance of the active site gorge here, which is represented in white on the blue surface of acetylcholinesterase. And it covers here the surface area, which is very big. It is more than 1,000 angstrom square surface, which is enormous surface of interaction. And this is maybe what we can expect from a very big peptidic toxin, but not very usual for example, organic inhibitor of acetylcholinesterase. So, the way fasciculin inhibits the enzyme was very interesting, and I do not have time to go into the details here, but that was very interesting to us to see whether intricate indeed the interaction were between the fasciculin side chains and the side chains of the surface of the peripheral site, but also to see that the fasciculin molecule was acting in a very simple way. Had a very simple way of action in contrast to what we expected initially when we thought that acetylcholinesterase had separate sites at the surface of the enzyme. And initially we thought that fasciculin was an allosteric inhibitor because we knew that it would bind on a site which was different from the catalytic site. And when this structure was solved, we found that the mode of action of fasciculin was much, much simpler than that. That was just a plug, a perfect plug, to plug the entrance of the active site and prevent the substrate to reach the active site at the bottom of the gorge. So, after this point, we wondered how other peripheral site inhibitors of acetylcholinesterase would be able to bind to the peripheral site of the enzyme, and whether they would bind to similar sites or subsites of the fasciculin binding site at the surface of the enzyme. And for this study, we used some peripheral site inhibitors, which were well known at that time, which were the propidium and decidium inhibitors, which are made of a phenylphenyltridinium moiety, which is connected with the tail. And the tail for the propidium is much smaller than the tail for the decidium molecule. And these molecules have interesting fluorescent properties, which were very instrumental for us to help us solve the structure of these complexes also. And here, you can see the structure, a close-up view of the structure of the propidium or decidium, actually. It is molecule bound at the peripheral site of the enzyme as well, and the white hole here is the entrance of the active site gorge. And you can see that the propidium molecule also binds to the peripheral site. And we also solved the structure of a galamine, which is a pyrogallol inhibitor with long tails here. And unfortunately, the crystal structure, the tails were too disordered that we couldn't see them in the structure, which means that the active part of the inhibitor is the cycle here of the galamine. And as you can see here, the galamine plugs the gorge, also the entrance of the active site gorge. But what I think is more interesting here is if we compare the crystal structures of these different inhibitors and the way they bind at the peripheral site of the enzyme, is that the phenylphenyltridinium of the propidium mimics in a way the determinants that are present at the tip of the second loop, the central loop of fasciculin. So these two different inhibitors of acetylcholinesterase have very common determinants for inhibiting the enzyme and plugging the entrance of the active site gorge. And the same for the pyrogallol ring of the galamine, which mimics a methionine side chain here, which is very instrumental for plugging the entrance of the active site gorge. And if you look at the surface, at the entrance of the active site gorge, which is covered with these different inhibitors, the biggest white surface is the one that is covered by fasciculin. And here is the entrance of the active site gorge. And actually the surface which is covered by galamine is the brown surface here. And the surface which is covered by decidium or propidium is the middle gray surface here. And I think that what is interesting here is to see that the acetylcholinesterase peripheral site, as we used to call it initially, actually is not a single site, but it is a subsite for multiple loci for the different peripheral site inhibitors of acetylcholinesterase. And I think this is something which is important when trying to design inhibitors or ligands for an enzyme, is that to imagine that a surface site or binding site for inhibitors might be made by different subsites which can be used in combination together or just separately from each other. So afterward, we thought that we were interested in trying to understand how acetylcholine itself bind to the peripheral side, because at that time, we knew already that that acetylcholine in excess would act as an inhibitor of the enzyme as well. And the question was, how does it bind to the peripheral side and how does it work? And we were lucky enough to be able to look at crystal structures, a series of crystal structures on multiple different substrates or substrate analogs of acetylcholinesterase using mutant acetylcholinesterase, which was generated in Palmer's lab by Zoran Radic, which was a mutated serine, a catalytic serine mutated in alanine, so that we could use substrate in the enzyme without any hydrolysis of the substrate. And we were lucky enough to freeze, to observe several binding positions of the substrate at the entrance of the active site gorge. And here the molecule of the enzyme, and the active site gorge is here, the gorge is here, the entrance of the gorge is here. And if we use the molecule of acetylcholinesterase, you can see that we can have substrate molecule bound at the peripheral site, exactly where the tip of the fasciculin central loop binds, and other substrate molecules coming down into the gorge up to the catalytic site. And we also had some analogs of the product molecules, able to see in the active site gorge. And a very surprising feature which we were able to observe at that time was to observe substrate molecules bound in another place at the surface of the acetylcholinesterase molecule. And that was very unexpected. And we had that was very surprised because, we were not expecting acetylcholine to bind at the surface of the enzyme at that place. Except that, that place is what we call the backdoor region in acetylcholinesterase. And this is a region which had been suspected for some time at the time to have some role in the regulation of catalysis by the enzyme. So maybe there is something interesting here that should be studied further on. Going back to, for continuing with inhibitors of acetylcholinesterase that bind at the entrance and the surface of the enzyme, I'd like to mention works we made with antibodies directed against acetylcholinesterase. These works were not done with Palmer, but I think they complete nicely what we've done together during these years. And these antibodies were monoclonal antibodies generated the old way in the mouse ascites. And we generated the FAB fragments of these antibodies and co-crystallize with the enzyme and see whether they would bind, what place of the enzyme they would bind. And these antibody FAB 410 bind to the enzyme at the gorge entrance, exactly as fasciculin does. And actually, they compete to each other when we do binding assays. And this is very interesting because this antibody binds about the same way as fasciculin, and this is the binding surface of the antibody at the surface of acetylcholinesterase with the entrance of the active site gorge here and the peripheral site here. And this is where fasciculin binds in comparison, and you can see that this is very similar. And the mode of action is also very similar in terms of complementarity of charge between the antibody, because it is a cation, as fasciculin is, with a very electropositive surface, which binds to the electronegative surface of the peripheral site of acetylcholinesterase. So here there is not much very originality in this antibody also, but if you compare the way the antibody binds, the determinants at the surface of the fasciculin molecule and the determinants of the antibody to bind to the enzyme, you can see that there are lots of homology between the two molecules. So this antibody is not more interesting in terms of a binder because the affinity for acetylcholinesterase is not as good as the affinity of fasciculin. But I think it is interesting to mention it because antibodies are much more useful now in perspective of therapeutics agents, because you can just generate I mean, nanobodies, for example, or even cell-free antibodies now, this is the modern technology, and manipulate them much more easily as their potential therapeutic agent or pharmacological tools as the SNEX toxins, which are kind of difficult to express recombinantly. And just to mention another antibody, which is an antibody which was very specific for acetylcholinesterase, and very interesting, is that this antibody is also a monoclonal antibody. It inhibits the enzyme, but it doesn't bind at all at the entrance of the active site gorge, and it binds rather in another part of the molecule, which is here. And here, this enzyme is a tetrameric enzyme, and we have 4 FABs of this antibody bound to the enzyme. And if you zoom on one molecule, actually this is that one, on one subunit, and you can see the molecule of acetylcholinesterase with the entrance of the active site gorge, the peripheroinoic site, which is in pink here, and the antibody binds on the side of the enzyme, which is not at all the same place. And actually, what I find is interesting, it binds in the backdoor region, the very same region, almost in the same region, where I show you just before the 2 substrate molecule bound at the surface of the enzyme. So this kind of antibody, again a recombinant antibody, but we can maybe try to make some new technology antibodies to create new ligands, new tools, pharmacological tools, to study, I mean, the mode of action of this backdoor region onto the substrate catalysis by acetylcholinesterase, and maybe imagine some new therapeutic agents based on this, I mean, information here. I cannot talk of acetylcholinesterase without talking of work which we made with the click chemistry inhibitors generated here between collaboration with Palmer and Charles. And just to make a long story short, I'm sure you know about the click chemistry inhibitors, and they were generated using connectivity between individual inhibitors of acetylcholinesterase, which is a tacrine here, a tetraaminoacridine inhibitor, connected with a Zide terminated linker, and the very same propydium molecule which I showed just before, the phenylphenantridinium molecule here, connected with alkyne reactant after a linker. And as you most probably know, when these combinations of these kinds of molecules with phenylphenantridinium and tacrine molecules connected with linkers of different lengths, and with reactants, azide and alkyne reactants, so all of combination of possible situations, were incubating with the enzyme. I mean, actually the enzyme was able to create in situ only one inhibitor here, a syn-triazole inhibitor connecting the two reactants. So only a syn inhibitor was created in the enzyme and not the anti-triazole. So as pharmacologists, and we were interested in trying to understand using crystal structures why, how did the anti and the syn inhibitors were differing from each other, and why the enzyme would generate the syn inhibitor and not the anti-inhibitor. So we saw crystal structures of complexes of the two inhibitor, the anti and syn inhibitor. And to make a long story short, I mean, what we could see is that the phenylphenantridinium at the entrance of the active site gorge here, and the tacrine moiety is bound here on the bottom of the gorge in the active site. And as you can see, I mean, there is a very, very tight fit of the inhibitor within the gorge. I mean, the fit with the walls of the gorge is very tight, and all the side chains in the gorge are very, very nicely accommodate the inhibitor. I mean, from the top of it to the bottom. And what's interesting here is that the most interesting part is the triazole, which is located here. And as you can see, in the anti-complex, the triazole is located here, while in the syn complex, it is located at a slightly different position. And maybe this is easier to see things here, so that the triazole in the syn is down the gorge compared to the triazole in the anti, because of the motion of the side chain here. It tries to goes up into the gorge to let this inhibitor go down. And also, this shortens the length of the inhibitor, so that this phenanthrinidium here can intercalate between this side chain here and that side chain here, so that the fit of the inhibitor is much more better with the syn than the anti. So just for this kind of, in the same kind of story, we were interested also to know where, what would happen if we remove if we remove the side chain here, which makes the difference between the syn and anti inhibitors. And using tyrosine, alanine mutant generated by Zoran Rajic in Palmer lab, I mean, we were able to stop structure of the same inhibitors in the enzyme without the tyrosine. And in the absence of the tyrosine, which is in the middle of the Golgi, and which makes the difference between the position of the triazole and the anti and the syn, we're able to see that the anti and the syn inhibitors were able to accommodate different conformations in the Golgi of the mutant enzyme compared to the conformation in the Golgi of the wild type enzyme. And I think this is a very interesting demonstration of the flexibility of the Golgi of the enzyme of acetylcholinesterase and the way these very nice and tight inhibitors are able to accommodate within the Golgi to make the better fit in this. So we also had another story here with, again, the syn and anti inhibitors with a shorter chain here, and only 5 carbon bonds here compared to 6 carbon bonds here with the Y-type enzyme. And here again we have a different positioning of the different moieties of the inhibitors within the Golgi of acetylcholinesterase, and which are connected with a synergistic modification of the side chain orientations all along the Golgi of the enzyme. So I think these inhibitors are very interesting because they demonstrate very nice synergies between the occupation at the active site of the tacrine moiety and at the pass, at the peripheral site, by the phenylphenoltridinium inhibitor. And that all this synergies is driven by very nice coordination of the side chain motions all along the Golgi from the bottom to the top or from the top to the bottom. So if I can use, I mean, there's some, yeah, I still have some time, I think. The second kind of molecule we were able to study with Palmer and using toxins also was, I mean, not exactly the nicotinic receptor, because when I was in my PhD training, I used the alpha-neurotoxins from snake venom, covotoxin, alpha-bunderotoxin, and some others to study the nicotinic receptor and try to understand how some difference between the neurotoxin could be interesting for studying different subtypes of nicotinic receptor, for example, between the long neurotoxin, the short neurotoxin, and so on. So when I came to when I was during one of my visits in the lab, I mean, this is when I met, I mean, Scott and Ryan, sorry. And Scott at that time if I remember well, I mean, you were expressing the acetylcholine binding protein from Aplysia, while the only acetylcholine binding protein that was published at that time was the one from Limnea. Am I right? Or, yeah. So I'm not going to remind you what is acetylcholine binding protein, but just that it is a structural surrogate of the ligand binding domain of the nicotinic acetylcholinesterase, acetylcholine receptor. I'm sorry. And the Limnea species was the first one that was described by the group of Sigma teacher Sigma in the early 2000s. And this acetylcholine binding protein, as we said before, is a very nice homopentameric assembly of subunits. And these subunits are very nice surrogates of the alpha subunit of the nicotinic receptors. So this ACHBP, oops, I'm sorry, are also very nice functional surrogates of the nicotinic receptors. In terms of pharmacological properties, I mean, pharmacological capacity of binding the nicotinic ligands, nicotinic agonists and antagonists. And here are all the organic nicotinic ligands which were tested at that time, and with a small agonist and the big antagonist here, but also the nicotinic antagonists, the peptidic nicotinic antagonists, which were made by the snake toxins, and the snail toxins, and the Baglerin, which is another snake toxins. And I think Baldo is going to talk maybe of this molecule later on tonight. So when Scott at that time was able to compare the properties of the binding proteins from Aplysia and Limnea with using a range of molecules which are agonists and antagonists of various sorts. And when I saw that, I mean, I jumped, of course, as a toxinologist on that part of the table, which was the one that was most interesting for me at that time. And the question I tried to ask with this molecule is, are ACHBP also conformational surrogates of the nicotinic receptors? In terms of the way they are soluble molecules with homopentamers of molecules, I mean, could they also have conformational changes that would maybe relate them better to the nicotinic receptor? And in fact, the question was because at that time all the structures that were solved by the group of Sigma were structures of a supposedly apo molecule and of complexes of the molecule with agonists. And all these structures look just the same. I mean, there was no difference between the bound and unbound molecules at that time. And that was not very reflecting, to my opinion, I mean, what would happen in the presence and in the absence of acetylcholine. And we were expecting to see something like that. So the question was, would bulky competitive antagonists, such as the alpha-neurotoxins, for example, from snake venom, be able to induce, or maybe better freeze, another conformation than the one that was at that time available? And so, as a toxinologist, I jumped on the opportunity to try to crack a structure of a complex between alpha-carbotoxin, long alpha-neurotoxin with 3 figures, and ACHBP. And this is what the complex looks like, with the 5 neurotoxins inserted at the interface between the subunits. And the closer view here, when you can see the tip of the loop, here the central loop, which is inserted at the, between loop C and loop F at the interface in the subunit. And you can see that lots of interactions between the tip of the toxin and ACHBP here, and I don't want to go into the details for that because we do not have time. But I want to stress out the similarity between the binding mode of the alpha-neurotoxin and the fasciculin to their respective receptor, which is the nicotinic receptor, here represented by ACHBP, and to acetylcholinesterase for the fasciculin molecule. And these 2 molecules belong to the same structural family of 3 fingers toxin molecules. And I mean, they are very similar in that sense that they have 3 figures, but fasciculin is the left hand and alpha-neurotoxin is the right hand. And this is the first difference, because the curvature is not the same, and they do not bind the same to their respective receptors. And of course, the fold is the same, but the determinants at the tips of the loops are not the same. And this makes the difference between the fasciculin to bind the acetylcholinesterase and the alpha-neurotoxin to bind the nicotinic receptor. So, I mean, we have very similar toxin force and interaction means, but very distinctive suction determinants for the toxins, on the toxin for their receptors. So, I mean, in the line of this work, I mean, we were interested with Scott in particular to solve a range of crystal structures of the various antagonists and agonists to try to see whether with the aplasia ACHBP we would be able to see different conformational states of the ACHBP that would be maybe related to the functioning of the real full-length receptors, nicotinic receptors. And here the toxins we will use with cobratoxin, which I talked just about, conotoxin EM1, methylic aconitin, loberin, epibatidine, and from the bigger to the smaller. I mean, of course, they all insert at the interface between subunits, and they all, I mean, have very nice binding fit in the binding pocket for the acetylcholine between loop C and loop F. And this is not very evident here, of course, because, I mean, you can see, I mean, they just look all the same. But if you look closer to this structure and think we were able to extract 3 main lines of information from this series of structures. And one line of information is that the aromaticness for agonist binding is central to the subunit interface. And that was not very new because this is what TTS Sigma had shown before, of course, but that was a nice confirmation of the concept. And here is epibatidine, I think. And if you look here, I mean, you can see the very nice fit, I mean, how epibatidine, the agonist molecule, is nestled in the binding site where acetylcholine bind between loop C and loop F. And the second point of line of information is that the binding pocket extends beyond the agonist core to provide for some molecules additional binding subsites. And this is particularly true for the antagonists, because if you compare the way the methylic aconitin binds and conotoxin binds, I mean, they have a common binding site which is right in the agonist core. So this site is right here between at the interface between the yellow and blue subunits. But conotoxin binds toward the apical side of the interface, whether the MLA binds toward the membrane side of the interface. So it means that here at the interface between these two subunits, we have, again, 3 subsites for binding the different moieties of the antagonist and promote maybe different mode of action of this antagonist. So the information also that, I do not have time to show the detail, is that the additional binding loci, which you observed here for methylic aconitin and conotoxin, govern the subtype specificity for the antagonist, because they have some more modification or variability in the side chains here, and so that these different antagonists can recognize different kinds of receptors differently. So just to summarize this, and this is the picture that Scott showed earlier, and what we were able to summarize here is that we had lots of conformational changes associated with ligand binding. And actually this is what the apoacetylcholine binding protein, the position of the C loop in the apo molecule. And this is the position of the C loop on the agonist binding epibatidine. And the C loop wraps around the agonist, as I said before, just to clump it into the binding pocket and make the fit very tight. And the C loop is in open position in the presence of the big antagonist here. So I think what is interesting here is that the loop C conformation of the position is able to adapt to the shape and the size of the ligand. So it doesn't mean that the ligand dictates the position of the loop, but rather the loop probably opens and closes in solution, and that depending on the size of the ligand and the nature of the ligand, which is agonist or antagonist, it can get closer fit to the ligand. And I think that maybe you showed something very similar, Scott, which is the range of opening and closure. I lost my mouse. I'm sorry. Where is it? Between a complex of ACHBP with epibatidine, so the agonist, and conotoxin, the antagonist. Yeah, it should play. It does. Thank you very much.
You're welcome.
Okay. And you can see that the conformational change here, between the two. Can I play again? Oops. Okay, too bad. So the conformational change is what kind of important, and this is not at all what was observed before with the initial structure of linear acetylcholine binding protein. And just to mention work which was made in collaboration with Ryan at that time, and concerning the partial nicotinic agonists, and you mentioned them very quickly, but I want to do the same. And the molecules which were tried at that time, which were assayed with these molecules. And the question was, how do these molecules bind to ACHBP, and can we extract information on the structures of the complex to understand how they work? And the partial nicotinic agonists, are characterized by fractional efficacy versus the efficacy elicited by acetylcholine in opening the channel. And you can see that these range of molecules have very different fractional efficacities. And to make a long story short, of course, they bind exactly at the same time in the nest for the core agonist binding site at the interface between subunits. But what they do is they promote different kinds of interactions with the surface of the interface. And they have much more interaction, which since they are bigger than just the regular agonists, the partial agonists promote more interaction with loop F on the complementary face of the subunit of the interface, so that loop C cannot wrap on them and block them as tightly as it has to be with the traditional agonist. And this is a scheme with same kind of scheme with antagonists, where the antagonist is so big with lots of interaction with the two loops, so that loop C cannot shut down on it. So to maybe the last line, which I want to talk about, and I have 2 slides from now, is a work which we made in collaboration with Yordi Molgo and Armen Zakarian concerning the phycotoxins from I don't know how to pronounce, dinoflagellate organisms. And these organisms are very important because, I mean, they contaminate the coasts. I mean, there are marine organisms that contaminate shellfish, bivalve shellfish, so this is shellfish which we eat. And these shellfish contaminate also the fishes, so, I mean, there is a real risk of contamination toxicity by the consumer. And you can see that the increasing prevalence between, I mean, these two sides, and I mean, in 30 years, and it seems that keep increases, I mean, with time, and the red tides. So these organisms are responsible for the red tides, especially, and they kill fishes also, and they may promote some human illness. They are toxic also from aerosolized forms, so that if you stay in an area where there are lots of, I don't know the name in English, but when the water goes up, just this. Okay. Okay. So you can breathe the toxin because it comes to your face, for example. So this is dangerous for that. So we were involved in studying two types of toxins, two families of toxins. And the first one was the family which is called the spirolytes and gymnodimines. And these are the toxins. So I'm not going to go into details into these toxins, but as you can see, they are very complicated toxins, polycyclic toxins, with, of course, imine toxins, so that they can bind to the nicotinic acetylcholine receptors. And Jordi Molgo and collaborator had shown that these toxins were antagonist molecule for the nicotinic receptors. So actually, these two families of toxins were shown by Jordi Molgo and collaborators that they have very high affinities for the nicotinic receptors, but kind of low subtype specificity. So they were able to bind and combine to most of the receptor subtypes, whichever there were muscle type receptors or neuronal type receptors. And so with these toxins, we made lots of assays, and our collaborators made binding assays on nicotinic receptors expressed at the surface of HGK cells. And they made electrophysiology assays on nicotinic receptors transplanted into ovocytes. And we made binding assays also, Zoran made that on ACHBP, the ACHBP molecules. And in combination with that, we cracked crystal structures of the molecules with the ACHBP. And what we were able to see is the way how these toxins also very nicely, tightly fit at the interface between subunits. And again, the loop C, yellow, and the blue loop F interface here. And this is the way the toxin fit at the surface, at the interface of this between subunits. And the surface which is occupied by the toxin at the interface is in a different color here. And the points which were interesting here to see is that these toxins, again, like a little bit like I showed you with methylic aconitine and chromotoxin just before, these toxins extend their binding determinants in the apical and membrane direction of the subunits interface. And this is the way they promote very high affinity binding. And also the other information which we had is the way they interact with loop F is very low. I mean, they have low interaction with loop F. And this is, to our opinion, the reason why they have very low nicotinic receptor subtype specificity. The last one is penile toxins, which are also marine toxins. And these ones were very different, a little bit different in their chemical structures, but much different in their pharmacological action, because these ones had higher nicotinic receptor subtype specificity. And we came through the same protocol, binding assays with the nicotinic receptors, different subtype, binding assays with ACHBPs, and cracking crystal structure. And here you can see the way the toxins bind at the interfaces, again at the same place. But the difference compared to the previous toxins is that rather than extending in the apical and membrane directions of the interface, these toxins I'm losing my words, extend toward the outside of the interface. And this is here, the bulky ring here, which makes the difference between this toxin and the previous toxins. And this is presumably what makes the difference and provides them with a higher subtype specificity, because they interact most with the sequence variable loop F, which is here. So just a take-home message, and I'm done. Just very short one. So complex binding sites that encompass multiple subsites or loci to generate distinctive combinations of interactions and promote distinctive pharmacological effects. This is important to take this into consideration when we design new molecules which are expected to become therapeutic agents, I think, or more simply, new tools for pharmacological exploration of the receptors. And that structural comparisons reveal conformational changes that could not have been predicted by molecular dynamic simulations. And I'm sure that Andy will agree on that. I hope he will. And that toxins can inspire long-term collaborations in addition to new therapeutic agents. And this is my dedication to Palmer. So the contributors for these rules, and I showed some of the pictures. There were no names affiliated to the pictures, but lots of all these people are here. The most important of them are here. I'm not going to mention all of them, and I just apologize if I forgot some of them here. Some of our findings here. And I want to just finish this by showing just a few pictures which were taken after my sabbatical in Palmer's lab. And all over the years. So that one was taken in Chile in 2002. That was, which Scott, you showed previously, when we were writing this paper all together in Marseille in the AFMB lab, and that one at the same time. And here we were working at the neuroligin paper, David. So, and here that was in Russia. And here this is a picture which I selected from the Légion d'honneur 2 years ago with Palmer in San Diego. Okay, thank you. And thanks for the extra time.
Maybe we take one question while we're just changing over. Question from the audience? Over here.
Just a short question, if I may, Mr. Pascal. You mentioned that you used a mutant of acetylcholinesterase to study the acetylcholine binding. And the mutant you used was phenylalanine instead of serine?
Serine alanine. Serine to S2O3 alanine.
Oh, to alanine. Oh.
Alanine.
You're on extended, so once we bring this up,
It had no activity.
it should actually show up.
So that was more easy to soak acetylcholine.
It's going to come up in a second. I think it's just,
Thinking about it.
yeah, it's taking time. We have one more question I think about.
Just a quick question. In your survey of the acetylcholine binding proteins, receptors, channels, and acetylcholinesterase, have at all you looked at galamine? Galamines, we're particularly interested in galamine because it happens to be an allosteric modulator for the muscarinic receptor, but it binds weakly to the orthosteric site where acetylcholine binds. It also binds very well to the site above the orthosteric site called an allosteric site. But I know that it is capable of inhibiting acetylcholinesterase. In addition, I know that it's a nice blocker. So a channel blocker. So have you looked at galamine and how galamine binds and
So we looked at galamine only as an acetylcholinesterase inhibitor, not as a nicotinic receptor binder.
So I did some structure of that, but I don't think we ever published it. Probably running around somewhere. We could probably do that.
Okay, well, that's thank you.
It's actually a pleasure for me to introduce Bill because Bill was recruited as part of a package in the early halcyon days of the School of Pharmacy when there was generous funding for new faculty members, and Palmer asked me to chair a search committee to bring in a number of people in natural products and organic chemistry. And we recruited this dream team that included Brad Moore and Ted Malinsky and Bill Gerwick. It wasn't much of a search. Palmer told us there are these three guys and let's go out and hire them. And The rest is history.
Well, thank you, Andy. But Andy, I thought it was the white beard and hair that we had this sabbatical. It is a special honor and pleasure to be here and to honor you, Palmer, here today with all that's gone on and the talk that I'm about to give. And we titled it as Palmer Taylor, Scientist, Scholar, Mentor, and Dean. And all of those things are very true, plus more, as we've heard about. But I think what I have particularly liked about you is that you really embrace natural products. And from many things I like about you. But as we just heard in Pascal's talk with the list of agents that she was working with, they're almost all natural products. And when we look at the pharmaceuticals that are in the clinic today and count up those that are natural product or natural product derived or inspired, it's 50 to 70% of all of the agents we use have a natural product origination. So natural products, as they've been isolated and discovered by one means or another, have contributed enormously to our science and to our medicine. And it's in that context I'm going to give my talk today. I also, Palmer, I was interested just to take a look on SciFinder and see, what were the statistics here. And I was pretty impressed to see 529 publications come up after filtering out duplicates. And you have some 858 co-authors. And that just is staggering. 858 co-authors. What does 858 co-authors look like? That's what it looks like. No, it's a testimony to not only your mentorship, but to your collaborative spirit. And it's in that, as I was thinking about what I wanted to talk about today, I thought, here we've been here 10 years, Brad, myself, Ted, and Peter, that were hired under this cluster hiring, that it's quite remarkable to think of some of the collaborations that have taken place for all of us. And so I just point out here some of the really amazing collaborations that I've had over the last 10 years that have fostered and stimulated new science that I had no dream of when I moved here. And so it's been very, very exciting to be a part of that. So what I'm going to do is give 4 short vignettes of different projects that have occurred over the last 10 years and give voice to some of these collaborations. So one of these projects came from some collections we made down in Curacao in the Southern Caribbean, in the Leeward Coast, very nice mangrove bays. And in one of these bays there was some mooring balls with ropes. And my student, Joe Nunnery, was out there snorkeling, and she collected these, and they went into our system where we make extracts, and then we do some fractionation and evaluate these broadly for biological properties. And this particular extract was very, very potent as a cancer cell toxin, and so we were intrigued to isolate the active compound, and that's published, so I won't go through it, but what we found were these epoxy ketone type natural products that represents the warhead. It's a warhead that you saw in Brad Moore's talk with the epoxamycins and gave rise to this clinically approved drug carfilzomib. But these carbohydrates were distinct and different in this P2 residue having this rather curious sulfoxide or sulfone. And we were interested, did this have differing properties? We tested it with the National Cancer Institute and their 60 cell line assay, and it does. These compounds do show a different spectrum of activity compared to some of the other proteasome inhibitors that have this kind of epoxy ketone. But it was really a collaboration with Mike Gilson's lab, which was quite fun to start to get an image and a picture of what this molecule might look when bound. These epoxy ketones, they form a morpholine ring with the catalytic threonine residue of the proteasome, all 3 catalytic centers of the proteasome. And then this is the rest of the peptide section. So this is the residue with the sulfoxide in this case, which is making a very interesting new connection that's not seen with other proteasome inhibitors with a backbone glycine and H. And so a new hydrogen bond was deduced from this and gave rise to increased interest to make do some synthesis in this area, which I'll show in just a minute to explore that. But one other aspect of the project was then to engage with Brad Moore and a visiting postdoc he had, Daniela Trevali, and she was very interested in the proteasome, as is Brad. And so we did some enzyme inhibition studies, but we also did some X-ray crystallographic studies together. Basically, Daniela from Brazil assembled some of these compounds, jumped on an airplane, flew to Munich, and worked with the world's expert in proteasome structural biology, Michael Grohl, and came back with, I think, 7 or 8 X-ray structures from that. We still have some to publish. But this shows the natural product with that morpholine ring that's created. And what we're looking at is the superpositioning of carmophycin reacted with that threonine in all 3 catalytic centers of the proteasome. It has chymotrypsin, trypsin, and caspase activities. So we see that it lines up quite well throughout in all 3 sites, but the side chain, interestingly, makes different contacts. That's a hexanoic acid chain, very lipophilic, makes interactions with lipophilic surfaces that are different in the 3 different catalytic sites. That ultimately became important to us, as I'll mention in a minute. So there's been some nice synthesis in the area. In fact, complete the structural elucidation, including stereochemistry. We synthesized the natural products and confirmed their structure. And so with that synthetic route in hand, we were able to explore some of the impacts of, for instance, the hydrogen-bonding nature of that residue in the P2 site. Was this the epoxy ketone really important to the activity of these drugs? And could we start to do some initial structure activity? And so we made this limited set of compounds and tested them for cancer cell toxicity to a couple of different cancer cell lines, and found some that are ultra high potency. These are single-digit nanomolar cytotoxins by virtue of this proteasome inhibition. And so the natural products, as well as some that had a mono or dimethyl glutamate, as well as the methionine derivative. We had high potency and some others, some medium potency. So these are 100 nanomolar type compounds, including this one, which has a simple enone. This was the stimulus for another entire project with the Moore group, where we explored in great depth the nature of that inhibition by an enone of the proteasome, and it very curiously undergoes a catalytically derived hydroamination reaction equivalent, and that was quite an interesting finding. So this work here has then stimulated a second generation of compounds that have played around with the nature of the warhead. We now have compounds that are down in the picomolar range, and moreover, we found that with activity at that level, these become warhead molecules for potential antibody-drug conjugates. And so we've been working with a company up in South San Francisco to attach these to antibodies. Initially, we did it through the hexanoic acid chain, putting an amine functionality at the terminus, and found that compound was absolutely dead because of that negative interaction with the lipophilic surfaces, as we later found in the X-ray crystal structure. So we've made new connections through P2, the P2 residue, because that's reaches out and is exposed to solvent. And we don't have the full assay data back on those, but we're hopeful. So we're continuing to work as this very promising class of anti-cancer, anti-proliferative type agent. Okay, a second short vignette from the lab comes from our work in Panama. We've been working in Panama since 2002 or so through the International Cooperative Biodiversity Group project of the Fogarty Institute at the NIH. And it's partners drug discovery from nature along with infrastructure development in biodiverse rich but economically poor or challenged countries, as well as training and outreach and helping to develop value in the biodiversity that's so prevalent here. So our program really focused in on the national park system, one island of Cueva. We've had some outstanding discoveries from there, but the project I'll talk about today was from Portobello on the Caribbean coast. And this involves some wonderful interactions with the Vivian Hook Lab. In fact, Vivian and I have a joint student that we're mentoring together. He's a fifth-year student, Bailey Miller, who's really done a lot of the work that I'm going to talk about. Vivian, as you probably most of you know, is a protease person, and particularly these cathepsins L and B are the subject of much of the work in her laboratory, and they have various therapeutic applications. So we screened our library of materials, Bailey did, as well as other people in Vivian's lab, and we came up with a very exciting hit, a very, very potent compound in this material called guanidamide A. Guanidamide A, as a structure, is an N-dimethyl isoleucine with a hydroxy acid and another leucine, and then an extended residue here that makes this enone, and then this head group. So kind of a modified lipopeptide is the way I would describe it. We had encountered this and first discovered it and described it. Roger Langington, when he was a postdoc in my lab, isolated this compound and defined it. Structure, it was a micromolar inhibitor to the malaria parasite. The structure has intrigued a couple of groups out there. The Conroy and Stoltz group both have engaged in chemical total synthesis of the compound and studied it for its activity against Plasmodium further, and found, at least to some sensitive strains, that it's down in the nanomolar range. So that's intriguing, and we've also pursued it now for this cathepsin L inhibitory activity. What Bailey found is that it was very selective. There's a real paucity of selective inhibitors to these cathepsins L and V. Their function is really not well understood at all, their physiological function, but nevertheless, their IC50s are down in the with a 30-minute pre-incubation. The enzyme kind of self-activates. It's down in the single-digit nanomolar range, and shows quite good selectivity, 40 or 120-fold over the cathepsins B and B. And with the Andy McGavin lab, Aaron Friedman, his former student, we had really a lot of fun to explore the induced docking fit of guanidamide in the cathepsin L, and with an overall orientation in that binding cleft with that yellow color depicting the thiolate of the cysteine protease, that either head group bottom or head group top. And so they did some very nice calculations, of which is their expertise, and the head group top was the clear winner in terms of best fit and structure. So this was actually very useful to actually see this, and so Bailey has gone on to do some molecular modeling himself, and this combined with work with Aaron Friedman gave rise to a series of hypotheses about could we enhance this even further in terms of the selectivity and potency of these compounds. And so dissecting the molecule, from the molecular modeling, we saw a head group, we saw what we felt was the enone pharmacophore that the thiolate adds in at this beta position in sort of a Michael reaction, and then we have some specificity induced by this peptide tail. Paul Bedreau, a student of mine that's just finished and gone off to his postdoc, completed the chemical synthesis of guanidamide. We had, from Bailey's work and the rediscovery of guanidamide as a cathepsin L inhibitor, we only had maybe 1 milligram of compound, and it really limited the biology that he could do. So with that goal, as well as the goal of making more guanidamide A, as well as analogs, Paul undertook this chemical synthesis. It took him a little longer than he thought, but he marched through it. He used pieces of the Conroy and Stoltz synthesis, some reactions, so he found really didn't work well in his hands, and so he modified them, the ones that are in green. And so it's a hybrid synthesis that's actually quite good, and he's been able to make about 20 analogs now, based on the structural biology work with Andy's lab, as well as with Bailey Miller's independent studies. And the synthetic guanidamide was tested by Bailey in a series of enzyme kinetic assays. It's down in the double-digit nanomolar for the K in activation of the enzyme. I had some very nice kinetic analysis. It's an irreversible inhibitor, as you would predict from that mechanism of it, the thiolate adding in a Michael reaction. But we've also then designed these 20 or so analog structures. And as you can tell, in this case, we're exploring some lipophilic pockets that were not well explored by the natural product. And indeed, we see an enhancement of the Ki down to the 18 nanomolar range. We have another derivative that we're looking at some intellectual property for that's down in right around 1 nanomolar. So the structural biology has really guided the design of these, and we're very excited about that. So dynamite A is a really potent and selective cathepsin L inhibitor. It's irreversible. We have a total synthesis, and we're doing some further expanded biological testing, and the orientation of this in the binding cleft came out of that molecular dynamic modeling. I should say with the McCarroll lab, actually, and that group that's working in parasite biology, we found that this compound is very active in some of the parasites as well, and we're exploring that further. Okay, a third big net begins in Papua New Guinea. We've had a number of expeditions there over the years with two of my very good colleagues, Phil Cruz from up at UC Santa Cruz and David Sherman from Michigan, where we rent out an entire dive boat, about 12 berths, and we spend a couple of weeks out collecting marine samples. And I've asked sometimes, where's the best diving in the world? I would say Papua New Guinea, because it's just got this fascinating underwater geography, these vertical cliffs that go into the deep blue, teeming with life and very clear and warm waters. And sharks, and that's our little shark belly there. Turning inwards towards the reef, what one sees is that some of the reef holes are covered by a fine net, or sometimes a tube. And if you then disturb that, coming out of that tube will be this small shrimp, but that's quite a large claw. It makes a loud, audible clicking sound underwater, snapping kind of sound, and actually it can nick your finger a little bit if it gets you. So we have brought this cyanobacterium, that's what this is, back to San Diego, grown it in the laboratory, and it doesn't make tubes, and it doesn't make nets. It's actually being woven by this shrimp in a really remarkable symbiosis between shrimp and cyanobacterium. It deriving protection in terms of cover, and this organism being highly unpalatable to predators because of the presence of this very, very potent toxin. This molecule, aflatoxin, we've been studying it for a number of years. It was first reported in the literature by Brad's father's group in Hawaii, and his very tremendous student and now independent scientist, Hendrik Lusch. It has this overall cyclic structure, and we became very interested in the biosynthesis of this compound. By the way, recently it's been reported, and this came up in earlier today in one of the talks. It's an inhibitor, specific inhibitor of the Sec61 translocon, which is a novel mechanism of action and has cytotoxic action down in that low nanomolar range once again. So our interest evolved. Well, we've isolated other analogs in this area and have been exploring the potential therapeutic application in this area, but also the biosynthesis. And it's very interesting with this tertiary butyl group. How is that being formed? And we predict it's coming from methyl groups of acetylmethionine being transferred to acetate and then a ketide assembly. But there's another very interesting modification here of a methyl group that's at a position that isn't normal in polyketides. And so there was some interesting biochemistry to explore there. So we wanted to gain access to the biosynthetic genes to study some of these interesting and intriguing processes. To do that, we brought it into culture, as I said, and here under a microscope you can see what those filaments look like. They're, this is about maybe 10 microns across, so very, very fine filament. The cells are shaped like kind of fat coins, and they're encased in a sheath-like material. So it looks very clean, but it's not. And we saw that very dramatically when we stained it with DAPI, and we can see that that sheath material was just loaded with heterotrophic bacterial colonies. And as try as we might, we've never been able to remove those bacteria and have a viable cyanobacterial culture. We don't really understand that, but these cyanobacteria have particular microbiomes that are distinct to them and seem to be important, perhaps, to their survival. But it was actually Nicholas Engeney going over to the laboratory of Mark Ellisman's that took this picture that was, that's just an amazing photo. This SEM depicts then the filament encased in that sheath, and you can see the exterior of the sheath is just very mossy with heterotrophic bacteria. Interior of this is essentially an axenic environment. And it gave rise to the idea, could we reach in there and pluck out some of those very nice clean cells so that we could get pure cyanobacterial DNA to work with in terms of any genome sequencing and biosynthetic work, and get it free of any of these contaminating heterotrophic bacteria. And in fact, we were able to do that with Roger Laskin and his very talented postdoc, Thomas Ishoy, where it wasn't quite reaching in there, it was more squeezing out, but then sucking up with a very fine micropipette, cell by cell, and washing them. And it was done a couple of years ago, as you can see, as we used 454 sequencing. But we got some nice sequence coverage, and most importantly, we got a series of contigs that could be put together and stitched together with some additional work to come up with the biosynthetic pathway for this very intriguing compound, aflatoxin A. And so looking at that for just a minute, the T-butyl group is put together in a cassette. This is one of these biosynthetic gene clusters, as we were hearing from Brad. It's a collinear gene cluster, so the genes are A, B, C, D, so forth, and they're used in exactly that order to progressively build this molecule in an assembly line type process. But very intriguing then is that formation of the T-butyl group, and we can see the genes that are involved in these unique methyltransferases reactions. We can also see the beta branch reaction that introduces that other unusual methyl group I mentioned. And so there's several features of biochemistry that we're currently exploring. We've actually just confirmed from some feeding experiments with C13 methionine to these living cultures that in vivo, the substrate that labels these methyl groups, in fact, is the methyl group of S-adenosylmethionine. At the terminus, there's another interesting motif, that is, it doesn't terminate with a typical thioesterase that cleaves and cyclizes this molecule, but rather a new motif. It has been seen, but biochemically is not characterized. It's a truncated NRPS domain, and presumably the condensation domain of that truncated NRPS module is catalyzing this hydrolysis and cyclization, forming that final bond. Okay, this has led to, in the laboratory, some more newer and now common techniques, as Brad was talking about, genome sequencing approaches to access these biosynthetic genes and perhaps compound discovery. And so this is 3 of about a dozen cyanobacteria that we've sequenced the genomes of. Lena Gerwick is really in charge of all that aspect of our joint labs, with Tiago Leo being a wonderful Brazilian student who's done the recent assemblies of these, getting these down to just a few scaffolds in each case. There are about 8 to 9 megabase DNA microbial genomes, so they're very large. And they make, we know, some very interesting compounds, and we've previously characterized those compounds as well as their biosynthetic gene clusters. Well, a postdoc in the lab, Karen Kleinbrew, this last year, was studying these in a little bit more detail from the genomic information and realized that 2 that we had studied before for jamaicamide and quercetin had a sensor histidine kinase of very high homology to one another downstream from the biosynthetic gene clusters. And these gene clusters are expressed at very high levels endogenously by the wild-type organism. So she got the idea, maybe this was sort of like the golden key to turn on these biosynthetic gene clusters and regulate them, and could she look and find them elsewhere? And she did. She found it in one of our Papua New Guinea collections, the one that makes the aflatoxins. And she looked upstream and she could find a biosynthetic gene cluster, the product of which she did not recognize or could not see. But she could make some deductions, as Brad was saying, and we can read these gene clusters now with some level of accuracy, not complete, but some level of accuracy. We can see that there was a long-chain fatty acid that was being activated to enter into the biosynthetic assembly. There was some very interesting chlorination taking place. We thought it was at the terminus, some extensions of this, connection to an amino acid. Serine was what was identified from analysis of the adenylation domain. Reductive offloading to make the primary alcohol, and then some further modifications. So we predicted that kind of a structure, and then we used what has been transformative, really, to natural product science. And I won't go into detail, as I'm sure that Peter will do this tomorrow, or I hope he will, because this is a really big impact on natural products, is to take mass spec data and the mass spec fragments from LCMS data, use those to see that molecules, based on their fragmentation patterns, can be arranged into families of molecules. And that's what's being depicted here, is that individual compounds are a node, and their relation to one another is that edge between them, the thickness of which indicates the degree of similarity. So that's an example. Peter and I have a joint grant that one of the first things we did was to analyze every fraction and extract we have in our collection, some 3,500 samples. Paulo Zato is the chief person working on this project. And then constructing these molecular networks. That's sort of the universe of marine cyanobacterial chemistry. There's thousands of compounds there for us to work on. We have no shortage of compounds to work on. And we could add in metadata. We could add in here their origin of the samples, or otherwise we could add in bioassay data or a combination of things. And this can then be a very useful tool for de-replication of known compounds. We don't want to re-isolate something that's already been described. Locate new analogs, for instance, of aprotoxins, where we're very interested in structure-activity relationships, or new natural products as a discovery tool. And we can ground truth this with standard mass spectra of our known and purified and rigorously identified natural products, and see that these are particular families of compounds that we know, like the aprotoxins or the curacins. So, using this technology, we went into the molecular network for this P&G strain that had this unusual biosynthetic gene cluster, and we could find a cluster of compounds that had a molecular formula that pretty nearly matched that idea that we gained from reading the genes, including, and very importantly, some chlorine atoms, which are pretty distinctive by mass spec and in their incorporation in natural products. So using that information, now we had a signature we could go after. We could follow this signature in an isolation protocol, standard methodologies, a very effective way to get to purified natural products in substantial quantities. We could use rigorous methods to solve complete absolute stereo structures, and we use that method to deduce the actual structure of this molecule as shown. So what happens is we have, indeed, the activation of a fatty acid, and then this very interesting coordination, not only at the terminus, but also at the omega-7 position. So really intriguing chemistry taking place there. It appears to be a new class of radical halogenase that's abstracting H dot and replacing it with Cl dot. We and others previously reported that in another class of enzyme. This is a distinct class that we think works by that same essential mechanism. We then have the chain extensions, the addition of serine, reduction of serenol, and then its conversion to that, this methylation to acetylation. So looking at that structure, we've been finding a number of these acyl amides from cyanobacteria over the years, and we thought, well, that kind of looks like some of those others in which we've seen the relationship to anandamide, the natural ligand to the cannabinoid receptor. So we tested it for that type of activity, and indeed, it's actually quite an active inhibitor of, so an antagonist of the CB1 receptor. We don't, and it's selective for CB1 over CB2. So that completes the four little vignettes that I wanted to share, but I think I've selected those with particular attention to many of you here in the audience that we've worked with you over the last 10 years, and I think it's something of the culture that, as I said earlier, that Palmer's helped to create here at UCSD that encourages that kind of collaborative spirit and inspires us to do things that we didn't do before. So thank you, Palmer, for that. And I'll just finish with that final thought. It's an amazing university because of people like you.
We have time for a question or two. Palmer.
Bill, a general question would be, as you look through these compounds and you've got these epoxy ketones and these enones and whatnot, what fraction of the natural products do you think have a warhead and are reacting covalently as opposed to reversibly?
Well, that's an interesting question, and actually Chambers Hughes, and I think Chambers is here, there he is, he's exploring exactly that point from the perspective that while historically there was concern about irreversible covalent binding compounds, the drug industry has kind of stepped back from that and said, yeah, the covalent modifiers are okay, and particularly electrophilic ones. And so Chambers is developing some methodology to selectively pull those out of extracts, which could be a very, very productive way to go. In terms of percentage, it depends a lot on the class of organism and chemistry and so forth. We have a lot of cyclic peptides that I don't think have the kind of functionality that gives rise to covalent additions, but then other of these molecules, yes. So it's variable.
Along the question Palmer asked, do you do any filtering process like paints or REOS, what they call the rapid elimination of swells, or looking for some reactive moieties that will indicate that they are not so selective, these compounds?
No, our protocol at the present time, which gives us a lot of leads. We have actually more leads than we can follow up on, and usually we're, with the dereplication tools that we have now, particularly the molecular networks, I think we're quite effective at getting to new molecular entities that have biological significance. Is we take our crude materials, and before we do anything else, we do a fractionation of those materials. And then those are our primary units for biological testing. If we find something active in particular fractions, because we've been doing this for a while, we know that, well, that might be a fraction that has a nuisance compound, as we would call it. I think you call it SWIL?
SWIL.
I think I
The
understand.
Vertex company called it SWIL.
Okay. We would call it a nuisance compound that just hits, as an inhibitor, a pan inhibitor of all enzymes. And we're not interested in that kind of a compound per se. So, for instance, our most polar materials oftentimes have some phospholipids and glycolipids that are kind of detergent-like molecules. So, no, but I don't think another aspect is, some people are studying sponges. Particular classes of sponges have some nuisance compounds that are frequently encountered and really like plant flavonoids or plant tannins. You really want to get rid of those before you do your biological testing. The cyanobacteria, we don't have quite that problem. So I think it's taxonomically driven as well.
So one last question from Mike.
So you showed one of those really nice macrocycles and the genes that generate it, and you talked about the closure. Is it, what's the mechanism that makes it close cyclically as opposed to polymerizing or something? Is it sitting on an enzyme that holds the ends close? Or?
Yes, and that's exactly it. There's a templating that takes place. Actually, Mike Burkhart, when he was with Chris Walsh, did some beautiful work in that area where they use the templating features of a thioesterase to close synthetic macrocycles that they could design and then feed to the enzyme and get it to exact that chemistry, catalyzing the cyclization over other reactions, hydrolysis or polymerization.
So we have a little break before the reception and banquet, but let's thank all of the afternoon speakers.